project is, I'm the director of the Medicinal Germplasm Repository Project located at the North Carolina Arboretum. I'm working with the Bank Creek Institute. I'm also faculty at NC State. We're starting a new project here where we're developing the nation's first official medicinal gene bank for native medicinal taxa specifically, but we're also interested in foreign taxa also. Let's see, do I have to point here? No, it's fine, okay. It's fine. I'll tell you a little bit about the Bank Creek Institute, and maybe I should stand up so my voice will carry a little further. The Bank Creek Institute is an economic development organization coordinating cutting-edge research to investigate bioactivity in Appalachian medicinal herbs for their efficacy in human health and also commercial product potential. So there are three divisions of the project: medicinal plant conservation, economic development, and research. My particular focus with the gene bank is medicinal plant conservation, #1, and then also research with the medicinal plant, with the germplasm. Oh, it's fine. So the question I always get very quickly is, what is germplasm? And people never want to shake my hand. But all it is genetic material. Specifically, it's hereditary material transmitted from one generation to the next. And in this specific area right now, we're focusing on seed, long-term preservation of seed for conservation purposes. So then everybody wants to know what a gene bank is specifically, and the gene bank is simply the germplasm storage facility for the seed for conserving biodiversity and research. And what has happened with medicinal plants in the U.S. is that the Convention on Biological Diversity has greatly changed collection practices for medicinal taxa. We used to always go to tropical countries and screen flora there and for drug discovery and for new medicinals. And as a result, North America really has been unstudied for their medicinal purposes. The Convention on Biological Diversity has changed this because in 1992 we decided that if you go to a foreign country, take their germplasm and develop it into a drug, you have to give some profit back to that country. So that's changed collection practices, and now there's a whole lot more interest in collecting here in North America. And so the botanists aren't getting away with going so far away for their travel dollars now. So the mission of our facility specifically is, #1, it's conservation. It's conservation of medicinal germplasm through collection, acquisition, exploration, and long-term storage. So our main mantra is conservation, but we can't just do conservation. We can't make a gene bank and just store the seeds, which we would like to do for long-term purposes. There has to be some research purposes that we can write grants and keep ourselves funded. So we conduct a variety of germplasm research utilizing these collections and our lab facilities. So I have a background, for example, in chemical analysis of plants for their medicinal efficacy. So we are encouraging researchers to screen the plant material that we collect while we're preserving the seeds long term, to make it more practical. We also want to encourage the use of the collections for collaborative projects for biotechnology research, crop improvement, and product development. We also want to maintain the collection by establishing a database containing all of the information for each accession, including any study associated with the collection. And we also want to ensure that any germplasm used from our collection is accurately vouchered, so that if any publications result from our medicinal germplasm, they can be repeated in 10 years. What's been happening with medicinal study in the U.S. is that scientists are usually going into a grocery store and getting a product from the shelf and conducting studies that way. And in 10 or 20 years, those studies, none of them can be repeated, so therefore it's bad science. And the National Institute of Health no longer wants to finance any of these projects where you don't know what your germplasm source is, where you can't go back to that original population and get the exact same source. And what we found out in some papers that we've just published is that, for example, with Prunella vulgaris, we can take 3 accessions of the same species. Prunella vulgaris is self-heal. It's a very common plant, everybody probably knows it, or heal-all, it's been used historically. We found a significant antiviral activity with it. So when I was in Iowa working with the NIH, I came back here, collected 3 accessions, one from Cedar Mountain, one from Roan Mountain, and one from Bank Creek, all in the same regional area. We assayed them for their bioactivity, and they were significantly different. And so this means that, this is what we're trying to prove, that the source of the material used is incredibly important. And what we'd like to do is to take this collection and develop products that we think regional growers in this area would benefit by since land prices are so high for farmers here, they have a hard time competing with other growers. We'd like to use, what you do is you use a germplasm collection to come up with a great product that regional growers could grow more profitably than something that they just maybe order from a different state. We'd like to put a lot of science behind the choice of the crop. We think that's very important. So why did we pick North Carolina, Western North Carolina? Why here and why now for the gene bank? As you see on this legend, vascular plant richness of ecoregions, you can see that the Appalachian chain by far has the highest vascular plant richness in North America. This is an optimal area. If you see we're down here at the bottom of the red, that's the Appalachian chain. This particular part of the Appalachian chain remained unglaciated longer than the other regions. So as the ice melted, we were the seed bank that restored the northern portions of North America, which from a genetic diversity point of view is huge. So that makes this region incredibly important for us to go in and preserve the seed long term as quickly as possible. Also, this is another map showing you biological importance categories of North America. And again, you can see that we're there with Florida and regions of California. And this is biological importance based on a lot of features. This is plants and animals and ecoregions, et cetera, et cetera. But other reasons, we've been geologically stable longer than other regions. And also we have a larger variety of landforms, climate, and soils, making us an incredibly diverse region. Also, another reason, the North Carolina Arboretum is hooked up to Pisgah and Nantahala National Forest, which gives me permits to collect on over 1 million acres right there of undisturbed, well, not undisturbed, no way is it undisturbed. But anyway, it's a wonderful, compared to Iowa, this is an amazing playground. I always came here to collect in Iowa. But I actually grew up in Brevard and Hendersonville, so I came home when I came back. Also another reason, a real big reason that we need a gene bank right now for native flora, especially medicinals, is the zone creep we're all seeing. You know, the climate zone maps are all being reevaluated right now using the more recent climate data. And what we're seeing is all the zones are shifting north. They're calling it zone creep. And so what we need to do is we need to get into the areas that are not going to make that creep as quickly as possible. For example, the heath balds and the spruce fir zone. Those are the two community types in our particular region that need to be collected and preserved as quickly as possible. And we do have grant projects in to get that done. In addition, why medicinals, dietary supplement sales are huge. In the U.S. alone, about $5 billion. Drug development, also, we're interested in, if we found something that was an incredible hit, we would be interested in providing something that would be natural product based, but we would not necessarily want it to be wild harvested. We would want propagation protocols figured out so that it could be done sustainably. We're not strong proponents of unsustainable harvesting methods. This is showing you the national plant germplasm facilities in the U.S. I trained at the one in Iowa. And these are the specific methods that we are using for our medicinal gene bank. We're adopting the exact same methods that they use that are quite stringent. And what this means